So welcome again, everyone from the side of the data stewardship team, a small subgroup of the team being present today, being Vanessa Fuchs next to me, Jens just throwing in a single short intro here, but giving another presentation right after, plus me, and we would like to give you an introduction on our work as data stewards to support the bioimaging community in general aspects such as the help desk and also in teaching and training. So as you're all aware, but maybe as a short, quick introduction, bioimaging data is often a key underlying set of experiments, of measurements, but it's generating data that is at least two-dimensional, if not higher dimensional, up to 5D and beyond. Easily, it's being annotated, hopefully richly with metadata. All of this data has to be handled. Data sets can easily increase or be huge in size in the gigabyte to terabyte size, depending on imaging techniques and the experimental setup. Due to the variety of microscopic techniques, there's various imaging modes, modalities, different techniques that is great to make use of to answer your specific research question or that maybe of your users. But at the end, this leads to the fact that there's many manufacturers of systems, and this overall leads to the fact that there's a large variety of imaging formats that the scientists or you as imaging facility staff or whoever deals with your data has to handle and to process. And overall, this requires sophisticated tools, workflows, including teaching and training for you and also for the community of scientists. NFDI for Bioimage is one of 26 consortia of the so-called Nationale Forschungsdateninfrastruktur, NFDI. So now you all have heard that term. Keep it in mind. It's one of 26 consortia. It itself envisions data as a common good for excellent research organized by the scientific community in Germany. So it's a national DFG-funded initiative to support research data management practices and implementations. And NFDI for Bioimage particularly focuses on RDM in the context of bioimaging and bioimage analysis. As you can see here in that scheme, it's substructured into 6 different task areas, TAs 1 to 6, of which the first 4 are more technical, dealing with, for example, data formats, IT infrastructure, multimodal data and its integration, as well as bioimage informatics and analysis, while TAs 5 and 6 deal with teaching, community outreach, and also governance and coordination.
Yeah, I will take over here. And in between all these 6 task areas, there is the team of the data stewards. So we are 6 people or 7 people distributed all over Germany. So here you can see all our faces. So Jens, you know me, you have seen now, and Conny. And then we have Mosen, Ricardo, Xenia, and Max still in the team. And we built a network of data stewards being there for basically all over Germany, for being there for the whole community. And we are located also in different task areas, yes, but we also have different expertise. So here you can see a little list of all the expertise that we have. It's open data and open science. We have people working in IT infrastructure. We have various fields of research like biological, biomedical, biophysical, people working in light microscopy facilities, people knowing bioimage data handling, processing, analysis. Also experts in bioimage data and bioimage data formats and metadata formats and open sharing of data in the community repositories, and also teaching and training. So what I want to say with that is that we actually have a quite broad list of expertise that we cover. And we are connected not only via over all Germany, but also with the different task areas. So we are there to connect basically the community with the NFDI for bioimage task areas and to support the community. And for this, we also have a service in place, which is the help desk service. So if you scan the QR code here, you can also reach out there, but I will introduce it in a second. Yes, and we offer help with different topics that I will also introduce in a second. We are there to develop tools and workflows together with the community and basically for the community. We connect the experts, as I said, and yeah, we share training material and we also develop training material and offer trainings. So if you have scanned the QR code, which is also up here, and you are seeking for help, you will land on this page here in the internet, which is our help desk mask. There you can already type in several things. You can basically tell us about your problem, your question, or your issue. And then we have a MetaMost channel where we actually can distribute your request, and you will get an answer within 1 or 2 working days. After that, we have already decided who is going to take over your request, and then we start finding a solution. We do that together with you. So either it's super easy and we just point you to the solution, say, hey, you can look it there, and then you have your solution already. Or if it's a little bigger problem, we can also sit together and talk. And then either we find a solution together, or we need to open up a bigger community use case where we can then also develop a solution in the consortium. And if you don't want to use the help desk, you can also use ImageSC, the forum. And if you use the hashtag or the tag NFDI for bioimage, this will also end up in our mailbox. So we can also answer you on that. Our help desk is in place since March 2024. And wait a second. No problem. Okay, since March 24, we have gotten 38 requests, from which 7 are currently still ongoing. We have 8 closed. That means that there was no further need for our help, or there was no response any longer. And we have 23 successfully finished. And the topics that we have covered so far is data management plans and our research data management issues for grant applications, for example. So we also have there data organization and data management, questions regarding Omero setup, general usage, connection to electronic lab notebooks, usage for image analysis, basically connecting it with image analysis softwares, metadata annotation, data publication, and if you want, your individual requests. So I think if I see that list and the question that Thomas was asking just previously, I think we fit quite well the audience here with all the topics that we have covered. Yes, and I would like to show you a short example request, or in general, how we handle data publication, if that is interesting for anyone. So we received, for example, here, so this is Inga and Xenia. Xenia is a data steward, as I've introduced, and Inga is working in our project office in NFDI for bioimage. But before she was there, she also had the request of publishing her PhD data onto a repository and make the data fair, findable, accessible, interoperable, and reusable. And they had a first consulting meeting where Xenia was understanding the data of Inga, and yeah, from that she recommended a repository where this data can go, which in this case was Bioimage Archive. Yeah, so they worked out the data structure together, and the metadata and the format of metadata was explained so that Inga then could go home with her homework to organize her data and also collect the respective metadata that was required. In a second meeting, they were then also checking up if everything is fine, if the data structure is according to Bioimage Archive, if the metadata that is deployed in a file list for this repository is correct, if there were any issues with the upload. Sometimes there are, and of course, yeah, they checked the acknowledgement, because we as NFDI for bioimage would also like to be acknowledged for the work that we do. It's, I mean, we do it for free, but this is our payment basically. And then after that, they went for publication. And if you're interested how that looks, you can also check out this number here, this persistent identifier for the respective publication. Okay. Then another thing that I would like to share, because we are also talking about training material here, I would like to tell how we do it at the HHU in the Center for Advanced Imaging, where Tom and I are actually working. And our Omero workflow, I'm not sure if this is interesting for anyone, but I thought it is. That's why I wanted to share. Yeah, so if a scientist at some point in his or her career decides, I want to use a microscope, he or she approaches us and he or she becomes a Kai user. They make contact to us and they actually get also help by designing their experiment, but at some point they get the introduction to the microscopes by our imaging specialists. And there already is the first infiltration. There's the first mentioning of Omero. And after that, after the introduction, the person at some point logs into Omero and then we get a notification and then the system basically starts that I called it our data management team. So Tom and I are taking that over. We make contact to the specific user and offer an Omero introduction, which is a one-on-one session for 1 hour. And in that, there's already the first offer for an RDM session where we then, if the person agrees on doing that, then we sit together and introduce the person to FAIR, to naming conventions, to data organization, data management in Omero, metadata annotation, and REMB. So REMB is the recommended metadata for biological images. This is the template that we use for metadata annotation. And if there is still interest and time, then we also create a customized set of Omero tags and also a customized metadata template that the person then can take home. And while acquiring images, already noting everything down, and then annotation in Omero is basically done as soon as the person leaves the microscope. If that was too fast and you want to read more, we have that also on Zenodo. And one thing that I already want to mention, but Connie is also going to mention later on, for doing all this organization in Omero, we also use training material that was produced by the i3D Bio team. So information infrastructure for bioimage data that you can find here, which is basically the basis for the training that we perform at Kai. And with that, I would like to hand over to Jens.
Cool. Now, one click, once live. Not much. I think it's a bit of a show and tell. So basically, okay, all this help desk thing, it's all abstract. So I'm gonna run through one help desk request to kind of give you a better feeling of what we're actually doing with them. So we get approached, Remedy for All. It's one research group has a publication and says, okay, I have raw image data for it, and I think it's a good idea to also publish this somewhere publicly. So we have an initial meeting, and okay, basically what we've seen before, what kind of data do you have? I say, okay, we have, for example, 16 plates, a couple of positions, different channels, so a couple of thousand images. And then they said, okay, then we also have an analysis workflow that we ran all our images through, and we have the results that came out of this analysis workflow. And we also have the segmented thingies that we measured that the raw is basically, oh, that's quite something. Okay, cool. So then the first thing is, can we have an example data set of that? So we can actually get our hands on the data, which is the first hurdle for them to, okay, how can I share the one plate and how do I get it out of my system? Which is already quite interesting. Most of the time this works pretty fine because it's not too big. In this setup, they had a specific system they were using, Columbus, which is from Revity. I think it's a proprietary software attached to the was it Opera or Operetta, high content screening systems. So they were able to export it out of there. They were able to share it with us. Then we had a look. It's like, okay, internally talking with each other, where do we see this data set? It's like bioimage archive further or rather IDR. And then internally in the background, we already kind of pulled our strings and talked to IDR contacts like, is this something for you? Might this be fitting for you? And in the same time, said, okay, maybe we orient ourselves bioimage archive first. Then we would have to look up and would have to look at the data. How would we, for example, get the workflow out of there to somehow be reproducible? How would we get the segmentations out of there? How would we get all of the measurements by ROIs, stuff like this? So a lot of technical stuff, which leads to the next meetings, mostly with the researchers, with the original authors who know their imaging data, And then one or two IT persons in the background who are familiar with their setup, with the Columbus. And in this case, it was a bit tricky because we tried to get the workflow out of there, and it was in a very weird long string that may be machine readable by Columbus, but by nothing else. We tried to get something else, didn't work. So like, okay, we have what we have. It's somehow technically it's there, the exported workflow. We just put it there and publish it with the imaging data. Can we get the segmentations out? Because they segmented, they had some phospholipidosis marker, and they segmented and measured the segmentation basically. And then it took quite some digging and some backup call to Josh with his wealth of Homero knowledge to, in the end, find out, okay, there's no way we can get this out of the Columbus system because it's simply not there. Involved, I exchanged SQL databases with them to try and dig around. It was not findable. I can talk a bit about later in the one-on-one how they solved it, but it was just insane. And they said, okay, we can't get the segmentations out. Can we get the measurements out? Yeah, the measurements are actually in a quite nicely exported way there. So we have the images, we have the analysis workflow somehow, at least technically. We have the results in a CSV that is really is nicely readable and publishable. Then we tried to say, okay, now export everything. We know what we want, export everything with this kind of CSV setup, with this omissive setup. We transferred all of the data. We properly converted all of the omissive data into OMISAR because Bioimage Archive and IDR as well now, only taking OMISAR as file format. And then we sat together last time with them, said, okay, now we need a couple of things for RMB, for this metadata schema, that are not super obviously in your paper. And then we kind of puzzled everything together for them, uploaded to the Bioimage Archive, and now they have a published data set. And in the background, wheels keep turning, and we are gonna publish it into IDR as well. But Bioimage Archive was the first step. But it's always, what kind of data do you have? Cool, can have a sample. Technical questions arise. Maybe the authors, researchers can answer them themselves. Most likely not. Then you have the IT person in the background who has no idea of what the image data is looking like. They just know, okay, yeah, we have the database. Do you want it? Yeah, cool, thanks. And then you kind of figure out and try to export and convert and make it readable and fair and reusable. That's it. That's our job. Done. Next.
This is probably very likely a bit more complicated help desk request. There's also more straightforward examples, but it really depends on what your challenges are, what your data is, what is it you would need. And from there we go, basically. Okay, so I jump to another aspect. There was the terms of training materials. So also within NFDI for bioimage, we are collecting and providing lists and materials thereof, which is on one hand a database collecting links to materials that I shortly introduce. We share materials as a community on Zenodo, all kinds of different materials. I also shortly will go into this. There is, for example, lecture series, some of them from Robert Hase from Uni Leipzig and SCATS AI, giving full lectures, several series, sometimes several presentations, for example, as Jupyter Notebooks on GitHub. We also reuse and recommend materials that has been created by the community and closely collaborating consortia and initiatives such as this nice bunch of Homero training materials from i3D Bio created by Tom, Christian, who's not here today, and others involved. And we already do share and will share more extensively materials on the consortium website that Vanessa also recommended earlier, NFDI for bioimage.de. And I will jump to a few details. So the training materials database is available on GitHub. It has by now more than 800 links to training materials that are being sorted depending on the kind of material which it is. It can be searched by keywords or texts. It's largely taken care of by Robert Hase and team. And the website is also open for contributions. So if you have materials that are being created that fit into the topic, please feel invited to also check this contribution information on how you could share the materials that maybe are not yet listed there to make the database more extensive. We also share materials on Zenodo. Currently, we have more than 100 records at current date. It's material that is mostly derived from the consortium directly, or it's derived from cross-consortia work, position papers, etc. And just listing here on the right the 3 most recent entries gives a good overview of the materials that we have listed there. For example, this includes the description of a tool from Thomas, the most recent one. There is a presentation of NFDI life science consortia combined together, so you won't see any NFDI for bioimage authors amongst the first names here yet. And there's also a slide presentation of basics of research data management by Robert, for example. And just covering a few more, showing you the scope of topics. There's best practices. There is visions and details of bioimage data formats from Josh. There's materials of bioimage analysis workflows. Here's an example from Ricardo presenting a Galaxy for the analysis of high content screening data. So you might also search there and check what kind of material there is available. And of course, get in touch with us if you have anything that would also fit in there, or maybe derived from this material, we can move forward. You can say, well, this is a good basis to get started, but could you help us maybe extend the workflow, or how can we implement this into our project? And with this, we come to the last slide, trying to summarize maybe a bit the role of data stewards. And this is nicely done by these great cartoons from Henning Falk. So on the left, you see basically a couple of, let's call them chipmunks. I'm not sure what they are. Basically not communicating because they all both come from their specific disciplines, talking different languages and trying to communicate, but in the end, it's not really productive and doesn't lead to much success. But if they find common language and try to communicate, and each of them approaching actually the other one, maybe they find a way to communicate and help out each other and creating some output, which maybe your scientific output at some point. And this is where we see our roles in trying to support this process of bringing different communities closer together and make them communicate in the end. And that's our part of the presentation. If you have questions, any feedback, any requests, anything that comes to your mind now or later, let us know. Approach us either here through the help desk address, or just get in touch with any of us.